So I'm going to be talking about newborn screening. So these are public health initiatives to test every baby, not sick babies, every healthy baby for a set of rare but serious conditions that can be treated. And the origins of newborn screening programs actually started here in the UK with the first screening tests for phenylketonuria, where a change in diet can prevent damage to the brain and nervous system. And these tests work by using both screening tests with high sensitivity, followed by a diagnostic test with high specificity, so we can identify as many babies as possible that have these conditions, but then also eliminate the false positives that come with those screening tests. And then once we identify those babies, we can apply the appropriate interventions. So today I'm going to focus on one disease, Pompe disease, which is one of the 40 conditions that is on the newborn screen in Washington State, my home state. This is an autosomal recessive condition. It is a glycogen storage disorder. So when you have a defect in the enzyme activity, you get accumulation of glycogen. There can be an early onset form, an infantile onset form that presents with cardiomyopathy and failure to thrive and respiratory insufficiency. And this form can be fatal without treatment within the first year or two of life. But it's treatable with an enzyme replacement therapy. And this treatment is found to be more successful if you can initiate before the onset of symptoms. So again, timing is really critical here. So the screening programs for Pompe disease involves an initial screening assay that's performed at our state lab. And if that screen is positive, if there is low GAA enzyme activity, there's a second tier assay, which is performed at our hospital in our biochemical genetics lab, which uses similar principles. And if that enzyme assay is also showing low activity, molecular testing is then ordered, which is performed in my lab by PCR and Sanger sequencing. But sometimes, depending on those results, we need to do parental testing to fully interpret the results. And our molecular testing is done on a subsequently collected blood specimen. So what I'm here to talk to you today about is the idea that we could use long-read sequencing and nanopore sequencing to streamline this process by capturing all types of the variants and being able to phase variants without needing to collect that subsequent variant from a parent. So I'm going to show you two examples today. The first one was a newborn with 2 blood spots that tested that showed low GAA activity in both that initial screening test, the second tier enzyme test. The testing in my laboratory, we found 2 variants. We found a heterozygous missense variant, as well as we're not doing systematic copy number analysis, but we are testing specifically for a deletion of exon 18 because it's a very common pathogenic allele. So in this baby, we found these 2 variants, but we don't know whether or not they are in cis or trans. So for this, we are using an adaptive sampling approach, which yielded high coverage over the GAA gene, and then we can look at this data and show that these 2 variants are in trans, and this information can help inform clinical decision-making. And so this is consistent with the parental testing for this individual. You know, we know this deletion of exon 18 is a common pathogenic variant. The missense variant was a more rare variant, but it had been seen in the infantile form. It's associated with a CRIM-positive status, which means this individual did not need immune modulation prior to the start of therapy. Next example, similar story. The first initial screening result showed low GAA activity from the dried blood spots, but that second tier test showed more normal activity. When we got a subsequent blood sample in my lab, we found 4 variants that met our reporting criteria. So we have 2 pathogenic variants and then 2 pseudodeficiency variants. And so these are variants that that show low activity in these in vitro assays, but in the body they function normally. And so the question here is, are these variants in cis or trans? What we really want to know is, are those two pathogenic variants on the same chromosome, or are they on different chromosomes? And we can do that with subsequent parental testing, or we can do it with long-read sequencing. So for this individual, we found that the two pathogenic variants were actually on the same haplotype, and the two pseudodeficiency variants were on the other haplotype. So this was a false positive result from newborn screening due to these pseudodeficiency alleles. And well, I think the timing, when we're talking about these life-saving interventions, this enzyme replacement therapy, that's really important. But this result is also really important. This is a family that was told you have a positive newborn screen for this devastating disease. Being able to tell them quickly that it is not, in fact, a positive result, it is a false positive, is also very important to be able to deliver that result, to resolve that, that anxiety and stress for that family with this newborn baby. So, again, this is some very initial data that has been generated in the research lab of Dr. Danny Miller, my neighbor at the University of Washington. But I think it's, it illustrates a lot of what we've been talking about here, is that the power of long reads to both phase variants without the need of subsequent parental samples to capture all types of variants. The GAA gene that I'm talking about today is pretty straightforward, but I think there are some other genes that are part of the newborn screening panels, like the SMN1 gene that's important in SMA that we know is really difficult, and we heard earlier in one of the breakout sessions about how nanopore sequencing has a lot of potential there. Another example is the ABCD1 gene on the X chromosome, where we don't do that testing in my lab because of the complicated pseudogene structure. That is testing that gets sent out. And so, like Dr. Mime was just talking about, one of the ways that we want to speed up this process is by being able to do more of this testing in-house in our laboratory without having to send it out. And by, as a clinical lab director, I think a lot about lab operations. When we have to develop specialized assays for different genes, that's really hard for a small staff. So I think one of the reasons that I'm really interested in nanopore sequencing is that we can use this technique and look at a lot of different rare conditions. And then some other exciting things that Danny's group is working on is whether or not we can do this on dried blood spot cards. Can we save more time by using a residual dried blood spot, and has some very promising data there. And I think looking towards the future, I'm talking about this as using long-read sequencing as that follow-up test. But there's some very exciting studies happening in the United States and in the UK and other places as well, as using genomic screening for healthy newborns, using short-read sequencing. But I think, you can see where I'm going with this here. I don't need to tell this audience. But thinking about the potential applications there for using long-read sequencing, where we could eliminate some of those false positives by knowing right up front that those two pathogenic variants are in trans. So with that, I'd just like to thank all of my collaborators, both at Seattle Children's Hospital and Danny Miller's lab. I hope Sunny Yi at some point will be able to tell you more about this story. She's a master's student in the Miller lab who is going to be really moving this project forward. But thank you for the opportunity to share these initial data with you here today. Thank you.